This talk is the penultimate talk in the 2008-2009 series of talks that, and other events that commemorate Darwin and evolution, because it's the 200th anniversary of Darwin's birth this year and the 150th anniversary of the publication of his most famous book, although he wrote a number of famous books, called On the Origin of Species. So, to proceed. Oh, and one last announcement. On Thursday, the ultimate talk for this series, or in other words, the last one, is Laura Landweber in CLAP 108, which is sponsored by the chemistry department. And she'll be talking about RNA and regulation of cells. Okay, so this is the title of my talk, and some brief conversation with Derek, who I thank for the introduction, convinced me that I needed to explain what I mean by utopianism. So here I provide a dictionary definition of utopia, either an imaginary and indefinitely remote place, I probably agree with that, often capitalized, a place of ideal perfection, especially in laws, government, and social conditions. And then lastly, an impractical scheme for social improvement. And then utopianism is the body of ideas, views, or aims of a utopian. So I hope that's clear.
Doesn't it mean no place?
Literally, yes.
Erewhon.
Well, utopia in Latin apparently can be interpreted as no place. But okay, so what is medical utopianism? This is my characterization. I wouldn't call it a definition because I'm sure there are problems with it, but it'll give you a sense of what I'm after in what follows. In my view, medical utopianism is a constellation of views constituting a mindset and characterized by the expectation, implicit or explicit, that new medical technologies will lead to inexorable progress in the preservation of health and to solutions for effectively all serious clinical problems. A critical and distinctive feature of this mindset is the disinclination to confront the ineluctable trade-offs, limits, compromises, and perverse or unintended effects inherent in new medical technology. So why give a seminar in this topic? Well, in my view, there's a need to counter what I would term powerful economic forces and social incentives that tend to favor excessive enthusiasm for new developments and trends in medical science and clinical care, instead of the academic ideal of thoughtful, dispassionate assessment of those developments and trends. Secondly, I would argue that maximally efficient use of available research resources, which until the stimulus package were considered somewhat limited, requires a balance between narrowly focused experimental activities, which most of us engage in and occupy the vast majority of our time and effort, and attention to the patterns of emerging insights across research communities and to the conceptual foundations of biomedical research. In other words, I think there's some value in taking a broader view from time to time. I'm not saying that you have to have separate human beings doing these two activities or that everyone should be doing the latter. But interestingly, I had just published a commentary recently in respect to a particular article in Science. I talked about what I called scientific hype. And one of the commenters wrote in, I think the science community needs to perhaps consider splitting doctorates into two forks. The first fork would be new research. The second fork would be mining, collating, and clarifying knowledge that we already have. I don't necessarily endorse the idea that you have to have separate tracks or that these activities have to be done by separate people. But I think it's interesting that someone else was thinking along this line. This is not someone I know or have any particular reason to believe is worth your paying attention to. On the other hand, I don't know that she's not. But I just think the second activity of mining, collating, and clarifying knowledge that we already have is often given short shrift. And quite frankly, the result of that can be wasting massive amounts of investment in projects that aren't really going to be very productive. There are several examples that come to mind, and we can talk about them later if anyone's interested. So here's what I'm going to speak about. I've already defined utopia and utopianism and medical utopianism, briefly told you about the motivation for the seminar. I'll give you some examples of what I'm referring to, talk about, in particular, there are other aspects of what I call medical utopianism we could certainly talk about, like some of the claims for aging that are being put forward by various people. But I'm going to focus on genome screening, the idea that we can scan the genome using very impressive new technology, identify disease-associated genes, and from those genes, develop new therapies. It's a nice idea. I suspect it will work some of the time, but I think it's been oversold. I will then talk about some of the realities of genotype-phenotype relationships and deal specifically with stochasticity, the randomness in the relationship between genotype and phenotype, and the degeneracy and structure-function correlations, briefly. I will then talk about some relevant history of disease-associated gene identification and therapy development, making the point that there already is a history of identified genes associated with particular diseases, and it's interesting to me how little account is taken of that history, which is well known and widely discussed. Finally, I will talk about some evolutionary considerations in accounting for complex phenotypes on the basis of genotypes, and then I will offer my conclusions. So let's go to the first concept. This is entitled, this is a surrealist painting by René Magritte, who's a Belgian famous surrealist painter. The title is Castle in the Pyrenees, but I think of it as a castle in the sky. So this is Lawrence Herkimer, and I facetiously ask, is he the patron saint of biomedical science? In case you don't recognize the name, and I wouldn't expect anyone would. I certainly wouldn't have if I hadn't seen his obituary. But he's the person who basically invented the modern concept of a cheerleader. And he was the one who patented pom-poms. Just so you have a sense of where I'm going. So here's an example of the kind of statement you'll find now in the public media. Ray Kurzweil is a very well-known inventor and technologist who's received all sorts of acclaim and awards for his software and computer-related inventions. And these are some statements he made to a publication by the name of Information Week, but I've seen him on television or radio or on the computer, seen him interviewed, and this is all in keeping with what he tends to say. In just 15 years, this is from 2006, in just 15 years we'll begin to see the merger of human and computer intelligence that ultimately will enable people to live forever. At least that's the prediction of author and futurist Ray Kurzweil. And it goes on to say, the inventor, author, and futurist predicts that in 15 to 30 years, nanobots will roam our bloodstreams, fixing diseased or aging organs. Computers will back up our human memories, and conference calls will be replaced by meetings in virtual resorts. I wonder if he thought about the possibility that that were true, then we would suffer not only from real viruses, but from computer viruses directly. Kurzweil isn't writing science fiction, so this article author writes. In fact, Microsoft's Bill Gates, a robotics director at Carnegie Mellon, an MIT professor, and a physicist have all endorsed his book. He has received these various prizes. So some people take him seriously. I am not among them. So what is going to happen in the future? According to Kevin Davies, who was an editor at Nature and wrote this book, Cracking the Genome, our children will be diagnosed for diseases they have not even developed and treated with drugs that match their body chemistry. Our grandchildren may be plucked from a pool of cells bathing in a petri dish after being screened for hidden flaws in their DNA. And our great-grandchildren will have dominion over the generation to come with the capability to engineer traits into genetic material, or the genetic material, as easily as sewing a button on a shirt. So that's quite a claim. Some of these things may in fact transpire, but they may not be quite as easy as sewing a button on a shirt or falling off a log. Here's another couple of quotes that are attributed to Craig Venter and Francis Collins from the same book. And this is from 2000 in the case of Craig Venter and in the case of Collins. Within 10 years, every baby born in a hospital in this country will have its complete genome repertoire determined. So that would be next year. That would be going on, if he's correct. And then Francis Collins said, in 10 years, we should be able to make predictions for you and me for what conditions we're most likely to be at risk for, and that in itself would allow us to practice some preventive medicine strategies based on our own individualized risks. He doesn't mention that we can do a lot of that already just on the basis of family history. And then here, Francis Collins gets even a little bit more utopian. Within 20 years, this is from 1999, so this is 10 years ago. Within 20 years, novel drugs will be available that derive from a detailed molecular understanding of common illnesses such as diabetes and high blood pressure. The drugs will target molecules logically as opposed to targeting them illogically, and therefore be potent without significant side effects. It's interesting that Francis Collins has a Ph.D. in physical chemistry, and I think this this idea that you can make drugs without side effects flies in the face of the fundamentals of physical chemistry, but we'll maybe discuss that later. By 2050, many potential diseases will be cured at the molecular level before they arise, although large inequities worldwide in access to these advances will continue to stir tensions. So what do I think about all this? Well, this is Isaiah Berlin, who was a famous historian of ideas, philosopher, essayist, and bon vivant, who was at Oxford for many years. And here's what Berlin had to say on the concept of utopia in his collection of essays entitled The Crooked Timber of Humanity. The notion of the perfect whole, the ultimate solution in which all good things coexist, seems to me to be not merely unattainable, that is a truism, but conceptually incoherent. It is true that some problems can be solved, some ills cured in both the individual and social life. We can save men from hunger or misery or injustice, but any study of society shows that every solution creates a new situation which breeds its own new needs and problems, new demands. And he goes on in a later part of the same book to say the following about those who seek utopia. Immanuel Kant, a man very remote from irrationalism, once observed that out of the crooked timber of humanity, from which the book title comes, no straight thing was ever made. And for that reason, no perfect solution is, not merely in practice, but in principle, possible in human affairs. And any determined attempt to produce it is likely to lead to suffering, disillusionment, and failure. So again, he's not against progress, and neither am I. The point is, if you expect to reach perfection and reach utopia, often the result is well short of that and worse than where you started. Now, in 1996, Edward Tenner published a book called Why Things Bite Back. This was about new technologies and what he calls the revenge effect. This included medicine, but was not in any way limited to medicine. But I'll just read you one quote from the book. The problem of today's medicine and main revenge effect of new therapies is that contrary to our expectations of technology, the more advanced it becomes, the more it demands in vigilance and craftsmanship. In medicine, the increased potential hazards of diagnostic and therapeutic equipment, complex procedures, and possible interactions of drugs require an unusual degree of attention. The proof is the surprising frequency of serious errors in medical practice. The study concluded, it refers to a particular study of medical errors, that 3.7% of all hospital stays include at least one adverse event. So the point is that as medical practice has gotten more sophisticated, that's all to the good in many cases, but there are perverse effects in that you end up with all sorts of errors, drug interactions that weren't anticipated, and so forth. So how should we approach then all these claims? Well, if one doubts that it's important to be skeptical in the, quote, real world, here's a quote from an economist, Alan Blinder at Princeton, talking about the current economic disaster. In the bubble era, even sophisticated people deluded themselves into believing that home prices would soar indefinitely and that lending risks were minimal. In a sense, they had assumed that utopia had arrived. On those weak foundations, a huge house of cards was built. Wall Streeters designed a hideously complex financial system to enrich themselves. Financial institutions took on far too much debt. People signed mortgages they could ill afford and did not understand. Regulators, the Bush administration, and Congress looked the other way. The bubble grew until it burst. Another example that many of you have no doubt heard about is the Ponzi scheme operated by Bernie Madoff, spelled differently, for about 20 years. And here's what I would have to say about why it succeeded for so long. His clients failed to ask questions about the fundamental nature of the pertinent activities. They just looked at the wonderful returns he promised them. They lacked skepticism about the claims for outcomes that were extraordinarily positive and regular in comparison to those to be expected in the light of past experience. And they fell prey to hero worship. And an example of that is I found this article in which it was quite interesting. Many of Madoff's investors have been left with nothing, having foolishly entrusted their life savings to a man they thought was God, as Elie Wiesel put it not long ago. That's a little bit much. Mr. Wiesel's foundation lost more than $15 million in the Madoff fraud, and he and his wife Marion lost their personal fortune as well. They really put all their trust in Mr. Madoff as if he were infallible. Now, this sort of hype is not new in medicine. It's been going on, and in some cases was much worse, because some of the things we're talking about actually do work to some degree. But here's an example that I thought might be of interest to people of Dr. John Brinkley. He wasn't actually a doctor. He never quite graduated, but he purchased a diploma from a diploma mill after spending 3 years at a medical school in Chicago without graduating. The remarkable events I'm going to chronicle here would likely never have unfolded in 1917 if young Dr. John Brinkley had not been hired as a house doctor at the Swift Meatpacking Company located in Kansas. And there he got to observe animals going to slaughter like goats. And apparently he particularly noticed the goats, as we'll get to. A couple of years later, after Brinkley had gone into private practice in Milford, Kansas, a farmer named Stitsworth came to see him. Stitsworth complained of a sagging libido, recalling the goats' frantic antics before they went to the slaughterhouse. The doctor semi-jokingly told his patient that what he needed was some goat glands. Stitsworth quickly responded, "So, doc, put him in, transplant him." Brinkley went to work, implanting a bit of goat gonad in Stitsworth's testicle. In other words, a xenogeneic orthotopic testicle transplant. Within weeks, the farmer was back to thank the doctor for giving him back his libido. And when his wife gave birth to a boy whom they appropriately named Billy, Stitsworth spread the word about Brinkley. Soon, Brinkley's business was booming. The testimonials poured in, and so did the money. Brinkley was charging $750 per transplant, and he couldn't keep up with the demand. So that's what's possible with word of mouth. Now, you've all seen headlines of this sort. Icelandic company says it has found osteoporosis gene. This is about some work at Decode Genetics, a legitimate company that does very high-grade genetic analysis and is particularly focused on the Icelandic population because it's relatively homogeneous and they have great records in Iceland. And so it says a gene linked to osteoporosis has been identified by Decode Genetics, et cetera. Of course, they call it the they make it sound like it is the osteoporosis gene or a osteoporosis gene. It is there to cause osteoporosis as opposed to just something that by chance happens to be a variation that is associated with osteoporosis. So can these genomicists look at the genetic egg, if you will, and see the adult phenotype from that? This is another Magritte painting called Clairvoyance. Well, here's a figure from an article by Ken Weiss, who's an anthropologist and geneticist at Penn State, who tends to be critical of various developments that he sees as overstated. And as he says in the accompanying text in the article, a consistent picture has emerged shown schematically in Figure 1. The poster child genes explain only a fraction of variation in a trait or a disease. So here are the number of loci, and here's the size of the effect of those loci on a given phenotype. And what has been found over many genome-wide association studies on all sorts of diseases, multiple sclerosis, schizophrenia, autism, type 2 diabetes, is that there are many, many loci at which polymorphisms have a small effect, and very few loci at which one or another allele has a very large effect. And Weiss goes on to say, most information gleaned from mapping consists of numerous small to very small individual effects. The most convincing genes have been found in more than one study, but usually confer relative risks on the order of 1.1 to 1.3, which for most complex diseases means small average absolute risk effects. The alleles of these genes usually have both low detectance and low penetrance. That is, the underlying genotype cannot be accurately predicted from the phenotype, and the genotype confers little power to predict the phenotype. So there's degeneracy in both directions. Alleles with higher relative risks are more likely to be replicated, but are as a rule too rare in the population to have cost effectiveness for variant-targeted therapies. So the common genes you might like to target aren't effective enough to necessarily think it's going to be worthwhile, and the very highly influential genes are too rare to make them worth a drug company's bother. So in a recent New England journal, in fact, it was rather convenient and fortuitous for me, last week the New England Journal published an article that had a series of commentaries on these genome scanning approaches. And David Goldstein is a very well-known geneticist at Duke who wrote one of the commentaries. And I quote from him, researchers now know how to properly account for most of the multiple hypothesis testing involved in mining the genome for associations And most reported associations reflect real biologic causation, but do they matter? Unfortunately, most common gene variants that are implicated by such studies are responsible for only a small fraction of the genetic variation that we know exists. So this is a geneticist involved in these kinds of studies as opposed to Weiss, who isn't directly. This observation is particularly troubling because the studies are largely comprehensive in terms of common single nucleotide polymorphisms, or SNPs, the genomic markers that are genotyped and with which disease associations are tested. We're finding the biggest effects that exist for this class of genetic variant, and common variation is packing much less of a phenotypic punch than expected. And here is a figure from his commentary showing for type 2 diabetes that the highest relative risk gene is a little over 1, 1.03 or 1.025 or something on that order. And the rest are all lower, barely over 1.005 relative risk. And if you look at height, there's this huge number of alleles which correlate with height. But the effect of each one of these SNPs is really tiny. There's another commentary in that very same issue of New England Journal by Kraft and Hunter. And as they show you in these figures from their commentary, there's a high degree of variability around the current estimates, talking about estimates of risk, given a current particular genotype. If there are 200 undiscovered locus associations, because again, we may not have found all of them, more than 7% of the subjects who are estimated to have triple the median risk of disease actually have less than the median risk. In other words, if you look at the, this figure shows you 3 curves. Subjects with 1/3 the median risk for that disease, subjects with the median risk, and subjects who have triple the median risk. And this line, this dotted line, this vertical dotted line, shows you the median risk. So all of the area under the curve for the triple median risk group that is to the left of that line are people who actually have less risk than median as opposed to 3 times the risk. This is another way of saying that our ability to predict risk based on these limited genotype associations is very unstable. That if you go to one of these gene companies that will tell you to send in a swab and they'll sequence your genome and look at, or look at hundreds of SNPs or millions of SNPs, whatever it is, thousands of SNPs, their risk predictions are not to be taken seriously. And as Kraft and Hunter go on, we are still too early in the cycle of discovery for most tests that are based on newly discovered associations to provide stable estimates of genetic risk for many diseases. Although the major findings are highly unlikely to be false positives, the identified variants do not contribute more than a small fraction of the inherited predisposition. And typically, like with height, the currently identified allelic variations that are associated with height variation account for, a small percentage of the total genetic variation. Now here's a slide that puts some of this in perspective. Since I am in the histocompatibility field, I have been aware of the fact that ankylosing spondylitis, a well-known and well-described rheumatic disease, is associated with one particular B locus allele called B27. And the relative risk is almost 90. Okay, let's go back 1 or 2 slides here. So remember, we're talking about a relative risk here for diabetes that as a maximum allele is under 1.3. And with ankylosing spondylitis, the relative risk is 90. Mohammed Khan, who's a colleague of ours at Metro and studies ankylosing spondylitis as well as suffering from it, had the following comment in a review he published just a couple of years ago. The remarkably strong association of ankylosing spondylitis with HLA-B27 was first described in 1973, over 35 years ago. Since then, a tremendous amount of literature has been produced, yet the issue remains as puzzling as ever. The epidemiological studies have shown that this association holds true almost worldwide and that AS prevalence in general mirrors the HLA-B27 distribution with peaks in those populations with highest prevalence of the antigen. HLA-B27 is directly involved in AS, but it seems to contribute only about 60% of the total genetic risk. So this is an allele with a relative risk of 90. And it still only accounts for a small portion of the relative risk. And we're talking in many of these common diseases like diabetes and hypertension about alleles with relative risks that are under 2, or at most 3.3 or 4. There are non-B27 genes both within and outside the major histocompatibility complex that also seem to be involved in disease etiology. I would also hasten to add that there is no therapy. Not only do we not know the pathogenesis more than 35 years after this gene-disease association, we have no therapy based on this gene-disease association. Now, another example of pharmacogenetics is provided by a test we actually do in our lab, the histocompatibility lab at University Hospitals, or University Hospitals Case Medical Center, as it's currently known. And this is a test for hypersensitivity to a drug used to treat HIV infection, abacavir. Apparently, it's a pretty good drug. Michael Letterman's in the audience. He can certainly testify to just how good a drug it is or isn't. Now, it turns out that people who are HLA-B5701 are hypersensitive to this drug. And if you have this hypersensitivity reaction, it's quite serious. It could be life-threatening. So it's something you wish to avoid as a physician and as a patient. So Simon Malal is an investigator in Australia who studied various aspects of HIV, and he published a paper in New England Journal recently about this particular test. And he says, given these estimates that 5.6% carriage frequency of B5701 in our study, the overall estimated positive and negative predictive values of HLA-B5701 testing for hypersensitivity reaction to abacavir in this population are 58% and 100% respectively. So in his population, the negative predictive value is great. If you don't have 5701, you don't have to worry about the hypersensitivity, although it may not actually be 0. At least in his population, it effectively was. But the positive predictive value is terrible, 58%. So we do this test, and it's probably worth putting these patients on a different drug, assuming there is another drug that's of comparable therapeutic benefit. But again, this is a very impressive association, but its ability to call whether you're actually going to get the outcome, the ability of this allele to predict whether you're actually going to get the hypersensitivity if exposed to the drug is not very impressive. Now, here's an example of the lengths to which this craze can be taken. Some time ago, the polymorphism at the alpha-actinin 3 locus was shown to be associated with performance in sprinting and powerlifting. And this gene is expressed in fast-twitch muscles, and it was discovered that 50% of 107 elite white sprinters, although in the current era, I don't know how many sprinters who are elite are white, but 50% of them had 2 so-called R alleles. No elite female sprinters had 2 X alleles, which are the, quote, slow alleles. And all the male Olympians in power sports had at least 1 R allele. Therefore, it made sense to a company in Australia that they should do a test for $149, and another American, and it's being offered in the US through another company in Denver. And there's a separate company in California that's going to charge $399 for you to test your little kid and find out if they're going to be an Olympic sprinter or a power lifter. So here are a couple of quotes from an article about this in the New York Times. When Donna Campiglia, someone I don't know, learned recently that a genetic test might be able to determine which sports suit the talents of her 2-year-old son, Noah, she instantly said, where can I get it and how much does it cost? In a health-conscious, sports-oriented Boulder, this company is playing into the obsessions of parents by offering this test for $149 that aims to predict a child's natural athletic strengths. The process is simple. You swab inside the child's cheek and along the gums, collect the DNA, return it to the lab for the analysis of the ACTN3 gene, etc. And the test goal is to determine whether a person will be best at speed and power sports like sprinting, football, or endurance sports like distance running, or a combination of the two. And it refers to a particular study that links this polymorphism with athletic ability. Now, the article goes on to say, some experts say this test is in its infancy and virtually useless. Dr. Theodore Friedman, who's a very well-known and respected person in the gene therapy world, the director of the University of California, San Diego Medical Center's interdepartmental gene therapy program, called it an opportunity to sell new versions of snake oil. This may or may not be quite that venal, but I would like to see a lot more research done before it is offered to the general public, he said. I don't deny that these genes can have a role in athletic success, but it's not that black and white. And in fact, the person who published or one of the authors on the 2003 study dissociated himself from this whole enterprise. So what the people offering the test for $149 or $399 don't mention is that they found in a Spanish long jumper, who's obviously a talented athlete, it was Olympic level, he had none of the R alleles. So somehow he managed without those particular alleles at that gene. There are at least 200 genes, apparently, which have been found to be associated with athletic performance. Maybe not all with sprinting or powerlifting, but just to keep in mind that there are probably lots of other important loci. And here's the point I would make to any parent who wants to spend $149 for this test. You can assess the phenotype for free, and it can be repeated at will, and it takes account of all the genes as well as environmental factors. If you want to know how fast your kid is going to be, take them out and run. Let them run and clock them, and then you'll know. It's more accurate, it's cheaper, and it's, as I say, infinitely repeatable. Now, here's a comment from Dr. Goldstein, who we previously quoted in an article by Nicholas Wade in the New York Times. The unexpected impasse also affects companies that offer personal genomic information that had assumed that they could inform customers of the genetic risks for common diseases based on researchers' discoveries. These companies are probably not performing any useful service at present, said David Goldstein, who I already introduced to you. With only a few exceptions, what the genomics companies are doing right now is recreational genomics. The information has little or, in many cases, no clinical relevance. Another point I wanted to make is based on the following. This is a gene screen result for lupus in mice. This is a mouse model, not the human. And what you can see is that there are a lot of loci which have been identified and associated with lupus in mice. So these are loci at which there are one or more variants which are associated with a predisposition to the autoimmune disease. Now, in my own work, when we knocked out a heavy chain constant region gene in the immunoglobulin heavy chain complex on chromosome 12 in the mouse, we found that those mice lived a lot longer in a background predisposed to lupus than heterozygotes and wild types, which are these two curves. And the point I want to make is that the locus involved is here on chromosome 12. And the only other loci known to be involved with lupus on that chromosome in various mouse strains are much more centromeric in their location. And so the point is that this gene, the gamma-3 constant region gene, doesn't vary in the inbred strains of mice. So the reason I bring this up is simply that the focus is entirely on gene products encoded at loci that vary in a way that associates with presence or absence of disease. What that fails to take account of is that sometimes gene products at loci that don't vary are extremely critical in the pathogenesis of a disease. And most of the therapies we currently use probably target loci, or the gene products, I should say, of loci, that in fact aren't identified necessarily as varying in association with disease prevalence. So why is it so hard to associate genotype with phenotype? And now I'm going to spend a little time talking about a couple of issues that may help us understand that. It's not a comprehensive presentation that would take way too long. So this slide shows you a paper from the Elowitz lab where they looked at cells expressing two different types of GFP, different colored GFP-like proteins from a given gene promoter, and found that over time there was a lot of dispersion in the relative amounts of the two genes that are being encoded by different loci. And these are presumably otherwise genetically identical cells. And what this is showing you is that over time they begin to develop essentially different phenotypes purely on the basis of randomness. And here are some of the slides which illustrate this effect where you get these different colors all mixed in together. These are cloned cells, effectively. I think these are E. coli. And what they're showing you is that if there are differing amounts of the two GFP-like proteins, you get different shades of green, yellow, orange, et cetera. And so some of these cases, you see that sort of variation. Here's another paper, Non-Genetic Origins of Cell-to-Cell Variability and Trail-Induced Apoptosis. I'm not going to read this whole abstract. I just want to go to the final sentence, which says, the protein state is transmitted from mother to daughter cell, giving rise to transient heritability and fate, but it's transient. But protein synthesis promotes rapid divergence so that sister cells soon become no more similar to each other than pairs of cells chosen at random. And then they do some fairly complicated analysis I'm not going to go through in detail. And essentially what you can see here as a function of time, you can see that the scatter in the y direction, the y-axis direction, increases as a function of time. And they claim that this is accounted for, and you see the same effect on this figure. Essentially what they're claiming is that with time from division, sister cells begin to vary in phenotype on a random basis. These are all presumably genetically identical cells because they're derived from the same precursor. And this is a slide from the same paper just showing you that when you look at the variations in cell protein expression, they seem to be able to account for the variation in apoptosis that these cells undergo after exposure to trail and cyclohexamide. So essentially what they're after in this paper is why is it that some cancer cells that are presumably genetically identical or at least similar to other cancer cells in the same tumor, some of them die and some of them don't in response to chemotherapy. And the conclusion they're being led to is that a lot of it has to do with randomness. So the point is the phenotype is a function of the genotype and randomness. Now, there are a number of other examples of random processes playing a role in phenotypes that may surprise some. Here's a press release from a study that was done at Harvard, and it says that genetics may play a surprisingly small role in determining the precise wiring of the mammalian nervous system according to painstaking mapping of every neuron projected into a small muscle mice used to move their ears. Not a clinically relevant study, perhaps, in the sense that the phenotype is not one that most of us worry about, but it does make an interesting point. They go on to say that this is from the actual paper. These results show that mammalian muscle function is implemented with a variety of wiring diagrams that share certain global features but differ substantially in anatomical form. And this variability may arise from the dominant role of synaptic competition in establishing the final circuit. Another example is provided from this book by Mark Kirshner and John Gerhardt, two well-known investigators. It's called The Plausibility of Life. And they point out that the growth of the vasculature is similarly dominated by a random component. Even the arrangement of large vessels appears to be irregular. If you compare the veins in your own two arms or legs, they will not be mirror images in contrast to your toes, joints, or large bones. And they go on to say that even for very large coronary arteries, there's a lot of variation from person to person. And amazingly, there's as much variation in identical twins as in unrelated individuals. So even though genes are obviously involved in wiring diagrams and vasculature structures, the exact way they end up isn't totally determined by your genes. There's an element of randomness, and there's often competition within the body that leads to different outcomes with the same genes. Here's another example. This is the first cloned cat, which was called CC for carbon copy, as opposed to copycat, which would have been my choice. But even though they're similar, you can see that the coloring patterns aren't identical. Of course, this is a younger cat, so maybe he'll grow into one that looks exactly like that. But at the time, at least, his phenotype was different than the parents. So now let's move on to how we get another reason we get these degenerate relations between genotype and phenotype. This is from a series of quotes from Ralph Greenspan, no relation, who's a Drosophila geneticist out in California. Difficulties arise because of the circuitousness of the path from gene to phenotype, a problem that is particularly acute in the study of behavior. He particularly studies fruit fly behavior. Not only are behavioral phenotypes very sensitive to non-genetic influences, but also the highly interconnected network of the nervous system sets up an additional layer of complexity between the gene and the realization of the phenotype. Specificity has been the shibboleth of modern biology. The concept of molecular specificity for sequence and for macromolecular structure formed the basis of the molecular biology revolution during the 50s and 60s. During the subsequent decades, even before the techniques of molecular biology flowed into field after field, the concept of specificity dominated much of the thinking, as it still does. Especially in the study of mutations that affect development, neurophysiology, and behavior, it became standard to claim the specificity of phenotype as a justification for biological importance, even if such specificity did not stand up to closer scrutiny. The expectation of finding specificity was so strong that pleiotropy was sufficient grounds for dismissing the importance of a gene. In other words, if a gene had multiple effects or its mutation had multiple effects, it was assumed not to be really seriously involved in that process. The first signs of trouble came with the attempts to identify all the genetic steps in a developmental pathway. In analyses of the vulva in the nematodes Cenorhabditis elegans and the compound I in Drosophila, it became clear that whereas a few genes were indeed specific to a pathway, many other equally crucial ones were not. In other words, they were involved in multiple pathways, perhaps. If the orderly steps of the pathway analogy no longer present a viable picture, and if instead we must come to grips with a more diverse, interconnected, and non-exclusive view of biological mechanisms, then certain assumptions must be let go. One of these is that every gene, and implicitly every protein, or gene product in other words, has evolved to fulfill some particular function, sometimes referred to as the Panglossian paradigm. The widespread finding of pleiotropy militates against this view, as does the ability of biological networks to reconfigure and improvise many strategies toward the same end. An assumption borrowed from physics that also falls by the wayside is that we can treat subsets of any system in isolation and still preserve its essential invariant properties. The high degree of interactivity is revealed, for example, in tests of epistasis and global monitoring of gene and protein expression indicates that even subtle changes to a system can alter its properties. And in this next slide, this is from an original report from Greenspan's lab, where he looked at a gene called syntaxin and made a mutant, and he had the wild type and the mutant, and he looked at the hepastatic wiring diagram, if you want to call it that, between a variety of genes in these two settings. And what you can see is that by changing this one gene, the interactions of all the other genes are changed. And if there was the level of specificity that Greenspan was being critical of in those previous slides that I just went through for you, this wouldn't happen. What he's saying is that the epistatic interactions, the functional connections among genes, are much more plastic than the dogma would have predicted. So to summarize in schematic form what he's saying, I would argue, and what I've been saying in a totally different context, quite frankly, based on totally different observations, is that although it would be great if structure and function were correlated in this way, one structure correlated with one function, and one function correlated with one structure, the reality is unfortunately more challenging. I just want to bring home that point because sort of in analogy to the Genome Project, there has been a structure project of similar magnitude and ambition. And this is taken from a commentary by Greg Petsko, who's a very well-known structural biologist at Brandeis, talking about the so-called Protein Structure Initiative, which was designed to screen thousands of protein structures, determine thousands of proteins as structures. When I was in college, I had a roommate who was a little weird. Okay, more than a little weird. One night at about 2 a.m., the campus police woke me up to tell me that he was sitting in the middle of the main street of our college town naked with a blanket over his head. I walked over to him, called his name, and said, "Warren, why are you sitting naked in the middle of Prospect Street with a blanket over your head at 2 o'clock in the morning?" He peered out from under the blanket, looked up at me, and said it seemed like a good idea at the time. My guess is that whoever was responsible for the Protein Structure Initiative must have felt the same way. The PSI is a fancy name for the US structural genomics effort. The PSI actually has two incarnations. The specific goals of PSI-1, which existed from 2000 to 2005, were to develop methodology and technology to increase success rates and lower costs of structural determination, to construct and automate the protein production and structural determination pipeline, and finally determine unique protein structures, lots of unique protein structures. By 2005, it became apparent that most of these goals weren't being met, nor were they likely to be met in the near future. When it looked like the PSI wasn't going to be able to meet its goals, what did it do? It moved the target in. These goals are so squishy, it would be almost impossible to meet them, or for it to matter much if they were. And he went on to say that, in his view, the people who came up with the PSI thought it was a good idea, seemed like a good idea at the time. And he has a variety of reasons why this whole project is not worth the investment that's been put into it. First of all, it's based on building homology models, and he says these methods are not highly reliable. Oh, I have a typo there, sorry. They're not very accurate when the sequences are less than about 50% identical in particular. Small errors in structures, which are inevitable in any structure determination, have dramatic functional consequences. Not all protein structures are worth determining. And protein structure does not predict function with current levels of understanding. Now, let's talk about, let's say we have the gene that we want. What is the likelihood we'll get a gene-specific therapy? Well, here's a short list of some diseases that were studied with genes in mind, and these are the years in which at least one gene was identified in association with those diseases. I already mentioned ankylosing spondylitis in '73, the oldest of which is sickle cell disease, for which the exact mutation, position 6 in the beta globin chain, was identified in 1957 in Nature by Vernon Ingram, cystic fibrosis 1989 by Francis Collins and Lap Chi Sui, and Huntington's disease in 1993 by Hausman's group at MGH. Or no, it was MIT. Maybe it was MGH. I don't know. Somewhere in Boston. Bottom line is, you can see that these all go back some years. In no case is there a gene-specific therapy based on that gene identification. Well, let's look at sickle cell disease. James Van Niel from Michigan established the mode of inheritance of sickle cell disease in 1949. Linus Pauling demonstrated that sickle cell disease was associated with a physically altered hemoglobin molecule in 1949. Some people date that as the beginning of molecular medicine. And as I just said, Vernon Ingram identified the exact amino acid substitution. In an article in Science in 1999, Professor Strasser said, even today, our extremely detailed understanding of the molecular etiology of sickle cell anemia has led to new diagnostic possibilities, but has contributed only modestly to improvements in therapy. And I took, this is current therapy based on the latest edition of Harrison's Principles of Internal Medicine, 17th edition, somewhat later than the one I used in medical school. The most significant advance in the therapy of sickle cell anemia has been the introduction of hydroxyurea as a mainstay of therapy for patients with severe symptoms. Hydroxyurea does not target the beta globin locus. In fact, it targets a different hemoglobin locus, and it was discovered entirely serendipitously, as I understand it, by trial and error. The other factor is that many therapies turn out to have unintended consequences, and I'll go through this quickly, but here are a couple of, I'm going to go through a couple of recent papers. This would go back to Tenner's concept of a revenge effect. So people have been using antibodies to the VEGF receptor as a way to prevent angiogenesis, which supplies blood to tumors. And what has been discovered recently is that although anti-VEGF antibody does have some very positive effects, depending on the timing, it can also have rather perverse effects. And in this paper, this is an animal model study, but what they found was that the longer they went with the anti-VEGF receptor therapy, the greater was the total amount of tumor in the animal. So this is control, 1 week and 4 weeks of therapy. And essentially, these are different grades of tumor, from encapsulated to microinvasive to widely invasive. And you can see that as you go from control to 1 week to 4 weeks of therapy, you go from predominantly encapsulated to predominantly invasive. And here on the y-axis, you have % total tumor per animal. And here again, you have the same kind of problem where at each of these time points, 12 weeks, 13 weeks, 14 weeks, 15 weeks, 16 weeks, the control has predominantly encapsulated tumor. The anti-VEGF receptor or antibody treated have primarily invasive tumor. And you also end up with increased metastasis. So if you treat with VEGF, here's an example of metastasis in the liver. It's actually better quantitated here. Incidence of metastasis, this is the control, and this is with the treatment. So this anti-tumor therapy, which does have beneficial effects depending on when you give it and when you look, can have the perverse effect of causing more virulent tumor. And this is a similar finding with another measure of metastasis. Another surprising result was recently discovered with erythropoietin therapy, recombinant erythropoietin. So this is from a paper in Lancet some years ago. The benefits of radiotherapy for patients with cancer diminish when anemia is present. Correcting the anemia has been suggested to reverse this hemoglobin effect, thereby improving cancer control. And so they've been treating these people with recombinant human erythropoietin to correct anemia, improve quality of life in these patients. And they also thought that this would improve the radiosensitivity of the tumors based on some earlier work. However, the potential of erythropoietin to improve cancer outcomes has not been established. Therefore, we investigated whether E-potent beta could improve cancer control and survival of patients irradiated for head and neck cancer. And what they found was that the patients treated with erythropoietin actually did worse in this study. And there have been, I think, some follow-up studies which have, again, found potential problems. So it isn't that this isn't a great therapy. It's just that it's not as straightforward and universally perfect as originally thought. There are other revenge effects or trade-offs one can think about. Vioxx, which was pulled from the market by Merck some years ago, as you may recall, if you were following that story, because it caused heart attacks. The HIV-1 vaccine in the STEP trial, which increased infection rates, at least putatively. Antibiotic use leading to infection with C. difficile, causing terrible gastrointestinal problems. And infections in immunosuppressed transplant patients. These are all well known to clinicians, and I'm sure that any clinician could list many more. Here's what Joseph Goldstein, one of the few Nobel laureate geneticists, had to say about this trend. Like journalists, many prominent scientists became seduced by the euphoria of the biotechnology revolution. They made a number of bold predictions, which are listed in Table 1. Worldwide shortage of paper, that has yet to occur. Sexual harassment by robots, that has yet to occur to my knowledge. Drugs without side effects, that will never occur, as far as I know. Organ replacement with biologically synthesized cells, people are working on it. And individual genomes on compact disks. Maybe that's true for Craig Venner and James Watson. That's about it. And he goes on to say, in many of these predictions, the distinction between illusion and reality is blurred, and that is what leads to me to suggest that the biotechnology revolution is like the surrealist revolution in art, in which the practitioners, artists, or scientists express their creativity by living and thinking in a world of dreams and fantasy. The surrealistic style and spirit of biotechnology is brilliantly captured in many paintings done 50 years ago by the famous surrealist artist René Magritte. Some of whose paintings I've shown you. On average, one new gene is cloned and characterized each day. This is back in '99. I'm sure it's more now. One new biotechnology company is formed each week, but only one new recombinant drug is approved by the FDA each year. And interestingly, there was just an article in Nature pointing out that last year, in 2008, I believe, there was a single drug approved for cancer therapy. As many as 700 therapeutic products are currently undergoing clinical trials by 167 companies, but 16 of the last 18 drugs failed in phase 2-3 studies. So it's hard to make a drug that works and is safe and effective, et cetera. If Magritte, the surrealist, were alive today, he might represent the situation as shown in Figure 6. And as he says, as Goldstein says, cloning a gene is only the first step in producing a drug. Or another way he put it is, a gene is not a drug. So this is the famous Magritte painting. It says, this is not a pipe. It's a picture of a pipe. So why evolutionarily do we get disease? Well, Randy Nesse listed 6 reasons when he came here and spoke, and this is from a recent Lancet article. Faster evolution of pathogens than hosts. We all know about that with flu and HIV. Environmental change that outpaces our evolution. That's postulated to account for diseases like diabetes, hypertension, obesity. Path dependence of evolution and other constraints on selection, inherent trade-offs among traits, sort of alluded to that. Variants that increase reproductive success but impair health, at least eventually. Things that, in other words, genotypes that might be great when you're young and not so good when you're not young. And high sensitivity of alarm responses. So here's another way to look at it. For Mendelian diseases, usually caused by rare, strongly deleterious mutations, think sickle cell disease or some of the less common immunodeficiency diseases that are single gene diseases, a mutation selection balance model in which disease alleles are continuously generated by mutation and eliminated by purifying selection is plausible in most cases. Now let's talk about more common diseases. Several known or probable susceptibility polymorphisms for common diseases ostensibly depart from this model in that the allele increasing risk is the ancestral allele. In other words, it was well adapted to a previous environment, whereas the derived allele is protective. In other words, the new mutation. Examples include variants involved in biological processes such as energy metabolism and sodium homeostasis. The epsilon-4 allele of the gene encoding apolipoprotein E, ApoE, which increases the risk to coronary artery disease and Alzheimer's disease, and carries the ancestral allele of 2 common amino acid polymorphisms. Interestingly, a resequencing survey showed that the haplotype class defined by the derived allele was associated with an excess of singleton variants consistent with the expansion of this haplotype driven by positive natural selection. So the point is, this allele, this old allele, which was probably well adapted to our environment centuries or millennia ago, was in the population. It's widespread because it was adaptive, and now it's maladaptive. And a mutant in that gene sequence generates a new allele, which instead of being the cause of the disease is actually protective. And that's not necessarily as common yet. And so we end up with this result that is from Ken Weiss's commentary in Genetics, where as I alluded to earlier, the high effect alleles are rare, and the common variants, or I should say the high impact variants are rare, and the low impact variants are common. And that creates a bit of a conundrum. So, as Ken Weiss puts it, biological traits are built up over episodic mutation, are built up over eons by episodic mutation and duplication events. Their genetic architecture is not as internally homogenized as textbook polygenic models suggest. Basic functions such as core molecules and signaling or metabolic networks, rate-limiting genes in physiologic systems or core protein domains are phylogenetically deep and widespread. Subsequent components arise that modify but must be compatible with these earlier ones. While the predominant image of life is the Darwinian one of winner-take-all competition, the predominant nature of life itself is of cooperative interactions among multiple components, signals and receptors, proteins in each other with DNA, and so on. So it is no surprise that multiple genes inherently affect interestingly complex traits. There is reason to think that the coding regions of early developmental or hub genes evolve relatively slowly. Partly this is because such genes are typically pleiotropic rather than being evolved for some specific function. Mutations with large effect, almost always negative, may be quickly removed by the cold hand of selection, having little chance to become geographically widespread. However, most functional genetic elements do tolerate variation, and most new mutations have little effect either on phenotype or on Darwinian fitness. These points together comprise one of the underappreciated implications of gradualism, a cornerstone of evolutionary theory. Evolution, working through phenotypes but only indirectly on underlying genotypes, has led to the complexity that buffers organisms against devastating mutation. So it's interesting that in 2001, after the initial claim that the Genome Project had been completed, after the publications in Science and Nature of the initial genome sequence for the human genome, Francis Collins and a couple of co-authors published a commentary in the New Republic called "Have No Fear, Genes Aren't Everything." I think the timing is quite interesting. After the money had been appropriated, after the project had completed, he comes forward to tell us that genes aren't everything. Unfortunately, the new focus on the genome has left some people with the impression that DNA's power is perhaps too considerable, that genes are too great a factor in defining who we are. Perhaps we are just marionettes being tugged along by the strands of our DNA. Perhaps our lives are nothing more than a formulaic drama with a plot line that was finalized before our birth. Fortunately, 10 years of intensive study of the human genome have provided ample evidence that these fears of genetic determinism are unwarranted. What he fails to note is that even before those 10 years, these fears of genetic determinism were unwarranted. It has shown us definitively that we human beings are far more than the sum of our genetic parts. To put it starkly, we have seen nothing in recent studies to suggest that nature's role in development is larger or nurture's role smaller than we previously thought. So in other words, actually, the Human Genome Project didn't change our view. And he goes on to say, it turns out that every case of sickle cell anemia is not created equal. Even when patients have the same 2 copies of the hemoglobin S gene, the disease may manifest itself in different ways. And this, in fact, was pointed out some years before the Genome Project got in full gear. This is in part because a separate set of genes in the genome, genes that code for fetal hemoglobin, can counteract some of the ill effects of the adult hemoglobin S genes. Interestingly, hydroxyurea targets the fetal hemoglobin locus. In most people, fetal hemoglobin genes turn off a few months after birth, and the adult hemoglobin genes take over. But sometimes the fetal hemoglobin genes are leaky, and they continue to produce fetal hemoglobin even into adulthood. When people with 2 copies of the hemoglobin S gene also inherit leaky fetal hemoglobin genes, their sickle cell symptoms are usually much less severe, sometimes actually not even clinically relevant. And this is primarily in Saudi Arabs who have the fetal hemoglobin polymorphism that makes for more fetal hemoglobin production and consequently less adult hemoglobin and lower hemoglobin S concentration, which is critical. So sickle cell anemia, widely considered to be the classic single gene Mendelian disease, is not so clear-cut after all. So in conclusion, there is a strong element of utopianism in many pronouncements regarding medical advances in general and those relating to genetics and genomics in particular. The claims made in such pronouncements merit careful scrutiny and measured skepticism, as they tend not to take into account historical precedents regarding the likelihood of success or the probability of unanticipated effects. Genotype-phenotype relationships are typically complex, context-dependent, probabilistic, and flexible, reflecting selection for robustness of system function in the face of both genetic and environmental fluctuations. Multiple genotypes can give rise to comparable phenotypes, and multiple phenotypes can arise from one genotype. While identifying disease-associated genes can be beneficial for both diagnosis and therapy, it is only a first step, especially for therapy, down an often long and tortuous investigational path. identification of many disease-associated genes will not necessarily lead directly to the development of a therapy. Even for those that lead to a new therapeutic agent, many will fail in trials or prove to be complicated to use effectively. Evolutionary principles can account for the large number of polymorphic loci, with each risk allele being of modest effect, identified in screens for common diseases. And finally, an excessive reliance on genetic approaches may fail to give sufficient weight to the relevance of alleles at loci, which variation does not appear correlated to disease prevalence or severity. So in closing, let me just say that the myth of Sisyphus, the Greek myth about the former king of Corinth who was condemned for eternity to roll a boulder up a hill and then have the boulder fall down again, isn't just a myth, it's a metaphor. It's a metaphor for human medical aspirations. And I would modify the myth a little bit so that instead of him rolling a boulder up a hill and then rolling down the same hill on the same side and starting over from the exact same spot, we roll our boulder up the hill and it falls down the other side to a place a little bit higher than where we started, or we just have to roll it up another hill that's still higher because we've created some new problem for ourselves. So it isn't that there isn't progress, it's just that progress is not like falling off a log. And I think it's better to be aware of what's coming than to pretend that utopia is just around the corner. Thank you. Happy to take questions. Cliff?
Does leaky fetal hemoglobin affect the incidence of malaria in individuals with the homozygous sickle illness?
I've never seen a paper that addresses that issue. I think,
Not the incidence of.
yeah, I think part of the Arabian Peninsula may have been subject to malaria, although I think the climate may be too dry now. So I don't know that it's an issue in the current, I mean, certainly that latitude is a malaria latitude. So, but it depends on other factors like, you know, how high you are in the mountains, and then it's cooler and drier and so forth. So I don't know that anyone's ever looked at that. It would be quite interesting to see. In fact, I don't know that it's ever really been fully elucidated how the heterozygous malaria genotype actually mediates resistance to malaria. I mean, it's presumed it makes it harder for the parasite to infect the red cells, but I've never really seen a molecular level explanation for how some sickle hemoglobin in a red cell with some normal hemoglobin A prevents the malaria parasite from replicating as efficiently.
So you cited in Saudi Arabs, but is leaky fetal hemoglobin present in the African populations where?
Not that I know of. I mean, if it is, it's much less common. So the original observation that led to that understanding was a group of doctors that I think were in Saudi Arabia with an oil company. And they started treating Saudis, and some of them had sickle cell disease, but some turned out had the genotype but didn't really have clinically severe disease or were almost non-clinical. And it was quite an interesting finding. Yeah, Cynthia?
Gary Brittenham observed that in southern India
Oh, okay.
in the early 80s.
Interesting. Dave?
So it seems that an underlying theme of your talk is that hyping your findings is selectively advantageous.
Yes.
And, so in a sense, I guess, do you have any thoughts about how the overall community can be changed so that hyping results like this, which is, I think you've demonstrated, quite deleterious, can be modified?
Well, that's a big challenge. You know, the foundation for that process of exaggerating findings or exaggerating the importance of findings is multi-pronged. So you have scientists with their own individual self-interest sometimes involved, whether it's intentional or otherwise. Maybe it's just legitimate enthusiasm. You have institutions like universities which seek to publicize the findings that arise on their own campuses to maximum extent. You have the publications themselves which benefit from having the most important and most attention-grabbing headlines and paper titles. So I don't know that there's any simple, easy way. I guess did my impression, actually one of my slides is taken from, let's see if I can find it, Ken Weiss's review. And he actually, so this is from Don Quixote. So I did my Don Quixote imitation just last week, actually, and had a commentary published online for The Scientist, pointing out that a particular paper in Science had claimed to revolutionize our understanding of antibody recognition. And even though I think the paper is a high-quality paper in many respects, the claim they make for having overturned how antibodies recognize antigen, I think was grossly overstated. And I'm not expecting that it's going to change anyone's principles for operating at Genentech, where the paper was from, or at Science, where the paper was published, or at The Scientist itself, where the news journalist, the scientific journalist, basically took what the author said and what a couple of commentators said, who were similarly unaware of the previous literature, surprisingly, at face value. So, you know, I'm probably a little bit like Don Quixote tilting at windmills, but you know, I did my part there to try and make people aware of the fact that this was grossly overstated. I don't know how to do it, really. I mean, there's no simple way. It's something I'd have to give some more thought. But you know, the problem is it's why there is no utopia, because everyone has a different interest. If you look closely, self-interest diverge at some point. And even though on a population level, it's not a good thing, on an individual level, even an individual institutional level, it is hard to pass up the opportunity to make the most of what comes your way in the way of scientific findings or publications. But it's something worth devoting further thought to. Any other questions?
You know, that was great, and I loved your negativity about this whole thing. But there have been occasional genetic loci that have yielded a major intervention, and apart from being these rare high impact loci,
Right.
Is there any commonality to those that have turned out to be useful?
Which do you have in mind?
The only, I mean, to a man with a hammer, everything looks like a nail. So I'm thinking about CCR5 and HIV susceptibility.
Right.
I'm sure there are others.
Well, people point to macular degeneration. There's an association with complement that was not apparent from the standard kinds of studies. And then there's a cardiovascular association, I can't remember which gene, that apparently is significant in accounting for certain kinds of heart disease. Now, those are associations which have significant impact. They haven't necessarily led to a therapy. So I agree. I mean, my point would be that inevitably this approach will generate some valuable gene-disease associations, either for understanding pathogenesis, for diagnosis, but much more rarely for therapy. And if they happen for therapy, it's going to take a long time. And even if you think you have a promising drug in the pipeline based on a target, the odds are not really with you that it's going to succeed. Some will. It's not zero. I would never say that. But it's not going to be straightforward and easy. And what I don't like is by portraying it as if this is the royal road to successful therapy. And in fact, they often use the word cure. In fact, Francis Collins even talked about personal genomics leading to medical nirvana, a phrase I found it hard to believe. But the problem with that is not that we shouldn't do these techniques. So I'm not arguing that we should stop doing them. It's that it distorts the relative distribution of resources. Because there is no, these people are full of numerators, but not denominators. And they can't tell you how much would have come from the money that was spent on genome-wide association studies that cost millions of dollars each. If that had been put into individual investigator-initiated projects on genetics or on other areas, maybe we would have gotten as much for our money. Maybe not, but I mean, they don't know. We don't know, but they certainly don't know. And they act as if it's obvious that this has been a great success. And as Ken Weiss pointed out, along the same lines as Greg Petsko in talking about how the protein structure initiative simply moved the target when they didn't reach it. The genomics people do a bit of that themselves. And Ken Weiss pointed this out, that if they don't get what they wanted out of the Genome Project or the HapMap project, they point to what successes they did have and say, see, it was successful. And again, it was successful in certain respects. I mean, the Genome Project has been a great boon for evolutionary studies and for all sorts of basic science and for certain activities in medicine. But in terms of producing therapies, you know, so far it's been pretty modest. It may be better 10, 20, 30 years from now. I'm sure it will be. But you know, it's not necessarily the only way in to achieving a good therapy. And it's not guaranteed to work. Anybody else? Well, thank you all.